compounds that can potentially act as universal platforms for creating transport systems for delivering plasmid DNA to cell, which can be used for biomedical applications. Delivery of DNA into eukaryotic cells is called transfection. Sensibly, transfection takes place in 3 stages. First of all, this is the condensation or compactization of the DNA molecule. The second stage of transfection is the passage of the DNA transfection agent complex through the membrane. And the third stage is the release of the DNA molecule into the intracellular space. For calixarine molecules, it has been shown in the literature that they can condense plasmid DNA and
sorry, your presentation is not demonstrate another slide. Please resize it. F and 5. Yes, thank you.
Okay. And the structure of the resulting condensates depends on the functional groups of calixarine. In our work, we investigated pyrrolurine containing amide and ammonium fragments with 2 different counter ions, iodine and chlorine. This pyrrolurine is positively charged and can interact with negatively charged DNA. To study complexation by the NMR method, we used a DNA decamer molecule since this method imposes a restriction on the size of objects. To obtain structural information about intermolecular interactions, we used nuclear Everhouse's spectroscopy method. In the noisy spectra, cross peaks appear between specially closed atoms. A fragment of the noisy spectra is shown on the left side of the slide. Here, the blue signals correspond to the spectrum of the DNA without pyrrolurine, and the red-yellow signals correspond to the spectrum of the DNA and pyrrolurine iodine mixture in a molar ratio 1:2. The spectrum of the mixture shows intermolecular cross peaks. Between the DNA atoms and ethyl and methyl groups of pyrrolurine atoms. Which signals indicate the DNA binding sites. Based on the obtained data, maps of the binding sites were constructed for pyrrolurine iodide and pyrrolurine chloride. The binding sites are similar for pyrrolurines with both counter ions, and the interaction occurs along the minor group of alien nucleotide. Also, based on the experimental data, a model of the pyrrolurine complexes with DNA was built in Autodoc program. Another way to study intermolecular interaction is NMR diffusimetry. The cell diffusion coefficient is inversely proportional to the hydrodynamic radius of a molecule. Wherein for a system with fast exchange between possible states on the timescale of NMR experiment, the observed cell diffusion coefficient is the average of cell diffusion coefficients in free and bound state. The slide shows dependence of the DNA black squares and pyrrolurine open circles cell diffusion coefficients on the molar ratio of pyrrolurine to DNA. Data on the left for pyrrolurine iodide and for the right for pyrrolurine chloride. Both pyrrolurines have close hydrodynamic sizes and see the same cell diffusion coefficients for non-bonded molecules. Under the addition of first small amounts to pyrrolurine to DNA solution, we self-diffusion coefficients of both DNA and purulence decrease sharply, indicating an increase of wear hydrodynamic size. And then with increasing molar ratio still definite values, the self-diffusion coefficients and hydrodynamic dimensions of two components is both systems are rather close in this concentration. And what can be explained as wear complex formation. During further increase of pyrrolorine concentration, the self-diffusion coefficient of DNA becomes constant, which means that the complex is saturated with the guest molecules and additional amounts of pyrrolorine stay in a free state. This can be detected by increase of observed self-diffusion coefficient of pyrrolorine, which what in the framework of two-site model means the increase of its free portion. Some differences between pyrrolorines are visible from the self-diffusion coefficients data. The self-diffusion coefficient of pyrrolorine iodide takes a minimum value, which corresponds to the self-diffusion coefficient of the complex, while pyrrolorine chloride does not reach it. We suggest that a certain amount of pyrrolorine chloride remains unbound. The hydrodynamic radius of pyrrolorine DNA and resulting complexes calculated in HydroPro program from PGB structure agree well with the already calculated from the self-diffusion coefficients. This fact supports the proposed structure of the complex. Since pyrrolorine forms complexes with low molecular weight DNA, it makes sense to test its ability to compact high molecular weight DNA. For this purpose, PGP-N1 plasmid contains 4,700 base pairs was chosen. The IFM image of plasmid is shown on the left side of the slide. This is a typical image of a long circular DNA plasmid deposited onto the freshly clipped mica in the absence of pyrrolorine. Single plasmids are visible in the form of supercoiled structures with several self-interactions. Sometimes individual plasmids overlap each other. As can be seen from figures in the center, in addition of pyrrolorines at charge ratio 0.1, there are no significant morphological changes. However, an increase in the number of self-intersections of DNA molecule is observed, which can be considered as an intermediate stage of condensation. In the case of pyrrolorine chloride, at charge ratio 1, plasmid appear in incompletely condensed conformation with loops emerging from the dense nucleus. For a plasmid mixing with pyrrolorine iodide at the same relative concentration, only the dense aggregates are absorbed and the loops of individual double helices are not visible. The detected DNA condensation allowed us to propose that pyrrolorines can assist with delivery of plasmid into cells. Pyrrolorines as a gene delivery agent has been investigated in the bacterial transformation experiments. Transformation experiments is the gene delivery in prokaryotic cells such as transfection is a gene delivery in eukaryotic cells. Plasmid PGP-N1 contains the gene for resistance to the antibiotic kanamycin, where bacteria, Escherichia blue, used in the experiment are not resistant to kanamycin, whose in an environment with kanamycin only transformed bacteria can survive. Pyrrolorines with both counter ions and increased transformation efficiency. An increase in the transformation efficiency up to 400 to 500% for was recorded for pyrrolorine iodide in the until studied range of concentrations. Further rise in efficiency was observed for pyrrolorine chloride with increasing concentration, and the efficiency at charge ratio 5 was 700%. This slide presents the results of the work. Thank you for your attention.